I want to make sure to give you guys an overview. I don't want to make any assumptions. I want to talk about the fundamental things that I think everybody should know about this COVID-19 pandemic. I'm going to talk a little bit about where we've been this year so far, and I'll end a little bit on some of the vaccines and how we're going to move forward with fighting and combating this pandemic. I titled this A Year with COVID-19 because it was a year ago this month that COVID-19 was first identified. Um, and we are now pretty deep in the pandemic, but we've made quite a bit of progress as well. So coronavirus, we've heard coronavirus quite a bit. They typically cause respiratory infections in humans. The COVID-19 disease is not the 19th coronavirus. Actually, there are 45 different coronaviruses that have been described thus far. And then the name comes from this electron micrograph image of a coronavirus where you can see the virus and these kind of protein-like appendages sticking out from the virus. It looks a little bit like a crown or a solar corona, which is a solar eclipse, what you would see from the sun peeking out from behind the moon. So that's where the coronavirus name comes from. And like typical respiratory infections, the symptoms include coughing, fever, difficulty breathing, especially in severe disease. And then along with those symptoms, transmission is via droplets from coughing or sneezing, either from the nasal or the oral membranes, mucous membranes. And then of course you can contaminate surfaces with droplets that contain the virus, which can aid transmission and person-to-person spread. So although there are 45 different coronaviruses that have coronavirus species that have been described, there are some notable outbreaks. The first one in 2002 was the start of the SARS outbreak, severe acute respiratory syndrome, SARS. The virus is called SARS-CoV or SARS coronavirus. And so that was mostly localized in Asia. There were some cases that spread across the world, but it was a little bit less it wasn't quite a pandemic, quite like this COVID-19 pandemic, as I'll show you in just a moment. Another notable outbreak was MERS, Middle East Respiratory Syndrome. This was again localized in the Middle East. The virus then was called MERS-CoV or MERS coronavirus. And in that case, it seemed like there was transmission from camels to people. And again, it didn't get spread as widely as the current pandemic. So that was back in 2012. 2019 is when we first saw COVID-19. So COVID-19 is the disease. Which comes from coronavirus disease 19 being 2019. And then the actual virus that causes COVID-19 is SARS coronavirus 2. So you'll notice that those virus names are quite similar between the SARS virus, which is the SARS-CoV, and then the COVID-19 virus, which is SARS-CoV-2. The reason is that they are quite similar viruses, which I'll show just in just a second. Genomically, they look very similar, but there are some distinct differences between these two outbreaks, and we'll touch on that in just a moment. I just want to describe viruses briefly so that it's understood these are not living things, they are what we call molecular machines. And what that means is that they don't have to encode all of the things that a eukaryotic cell, for example, needs to encode to replicate on its own. So they can be very simple in structure because they can infect and hijack a host cell and use that host cell's machinery to then replicate, make more of itself, and continue spreading. Because they don't need to encode all of the machinery and molecules that a self-replicating cell needs to encode, their structure can be very simple, very basic. In the case of coronavirus, for the COVID-19 virus rather, there is a genome, it's an RNA genome that encodes all of the genes to make that virus. There's this envelope, this lipid bilayer that covers and protects that RNA genome. And that's that blue structure surrounding the yellow genome there. And then you have these spike proteins that protrude out from the envelope. The spike proteins are quite important for several reasons. One, because it mediates an actual infection. It mediates the entry of the virus into a host cell. So I have a picture here showing the virus here. There's the genome in the middle there, the envelope surrounding the genome, and these protrusions are the spike proteins. The spike proteins can attach to proteins on our cells or receptors on our cells, and that can mediate a process where the virus can gain entry into our cells, then use our cells' machinery to make more of itself. The spike proteins are also very important for what we call tropism, which describes the types of organisms or the types of cells within a multicellular organism that a virus can infect. So if the spike proteins can specifically bind to something on a human cell, then it can infect those human cells. If it can't bind to anything on those human cells, then it can infect those human cells, and it limits the tropism. In this case, we know that the COVID-19 causing virus probably came from bats. It's now in humans, of course. We've seen it in other animals, including cats and dogs. And that can be important because as we're talking about controlling transmission, controlling infection, we have to consider not just human to human transmission, but also whether animals are transmitting it between other animals or to humans as well. So that's all kind of part of infection control. So I talked about comparing the SARS outbreak to this now COVID-19 pandemic. And I mentioned that they're quite similar genomically. So on a genetic level, their genomes are 79% similar. So there is some difference, 21% is different between these two viruses, but they are quite similar. The spike proteins are quite similar as well. There's a particular spike protein domain that's 88% similar between these two viruses. And the consequence of that similarity in the spike protein means or it helps explain how these two viruses actually use the same host cell receptors. On our cells, they bind to something called ACE2 to gain entry into our cells. Both of them use that same receptor. I mentioned the differences. So when we look at the infection rate, the number of infections, the number of deaths, there are some distinct differences. SARS wasn't nearly as widespread, only 8,098 confirmed cases around the world during that outbreak. COVID, on the other hand, is 73 million and counting, so this is a widespread pandemic, of course. And then the mortality rate was also quite different. SARS, it was estimated to be about 10% mortality. COVID-19, on the other hand, is quite lower. 2 to 3% is the estimate for mortality if you get infected with COVID-19. So it seems like when you consider the genetic similarities and the genetic differences, that 21% genomic difference between these two viruses, for some reason, has helped the COVID-19 virus be more contagious, transmit itself much more readily than the virus that caused SARS. So speaking of transmission, we can track the virus around the world as it's jumping from person to person, from animal to animal. And like most viruses, SARS-CoV-2, which is the virus that causes COVID-19, it mutates very rapidly, very readily, very quickly. What we know is that a lot of those mutations don't seem to be very harmful or very concerning. They don't seem to have any real impact on human health or disease. But there tend to be certain what they call variants that seem to be more common in different regions of the world, and they can track those different uh, SARS-CoV-2 virus variants around the world and see which ones are popping up more readily in different regions. One particular mutation, though, and this is a mutation that's not surprisingly in that spike protein. Remember that spike protein is important for gaining entry into a host cell. There's a particular mutation in the spike protein called 614G, at the 614 position of that spike protein sequence, that seems to actually make the SARS coronavirus 2 more contagious. It seems to allow it to be transmitted much more easily. There's another variant actually that I just read this morning that seems to be alarming people, and again, making the COVID-19 virus more contagious. But the good news is that when we look at the sequence of those viruses, they don't seem to have any impact on the vaccine. It seems like the vaccine will still protect against those strains, thankfully. And they also don't seem to have any impact on disease severity. So they're just making, as far as we know, they're just making the virus a little bit easier to be transmitted from person to person. So some concerns. So I have a list of concerns here. A lot of these were big concerns at the start of the pandemic, and I'm going to highlight some of them that I think are of interest. So I'm not going to talk about all of these. But one thing at the very beginning of the pandemic that really stuck out to me was the kind of racial data, racial inequality data when it came to especially the mortality rate of people who were dying from COVID-19. And there's a website with lots of data. It's covidtracking.com/race, curated by The Atlantic magazine. Really, really well done. But they have a lot of race data. You can see it broken down by state in the United States. And one thing that they discovered was that there were a lot of Black Americans dying at a higher rate than other demographics in the country. And that seemed to be quite an alarming trend. But when we look in just a moment at some of the risk factors for severe disease or death from COVID-19, it might be a little bit more clear why there was this kind of racial disparity in terms of the death rate. So there are lots of factors that influence whether or not you're going to be exposed to COVID-19, whether you're going to have severe disease. Of course, the types of job that you have, if you're in the service industry or if you have a transportation job and you're exposed to lots of different people. So socioeconomics, chronic diseases. A lot of Black Americans tend to have higher rates of chronic diseases, which just make the COVID-19 infection more complicated to treat and can lead to higher mortality rates in Black Americans. And then I have a listing of needs. Some of these things have been addressed at the moment. Remember, though, at the very beginning of the pandemic, access to PPE or personal protective equipment, so gloves, masks, gowns, was a really big deal. I remember many of us in the Spelman biology department donated PPE, gloves, masks, and gowns to local hospitals because they were in such desperate need. It seems like maybe that has been addressed since then. Access to sick leave, allowing people who are sick to stay at home seems, that still needs to be addressed on a national level. And then in terms of the racial inequality data, there is a caveat to that. We have a lot of data and information, but not all states in the United States are reporting race-related data. Also, it's not necessarily reported at a county or local level. So you have to make some predictions and some kind of generalizations when it comes to analyzing some of that race-related data, which some of you might be working on projects using that data So keep those kind of limitations and caveats in mind. Another concern I want to highlight is the adherence to prevention measures, which has become a pretty big topic. Whether people are going to continue to socially distance, wear masks, it's become quite an issue of concern. Another prevention measure, though, that the Pew Research Center has been looking at are the vaccine adherence, I guess, the vaccine guidelines, whether people are interested, willing to take a COVID-19 vaccine. They have lots of data about this, very easy to access. This particular figure is showing over time in survey results how people decided whether they wanted to get a vaccine or not. So this first bar here is from May of this year, May 2020. 72% said they would probably or definitely get the vaccine. That dropped down in September to only 51%. And since November, the November survey showed that kind of bounced up to 60%. So it seems like there's still, some concern and fear about the vaccine, the speed of the vaccine. And I'll talk a little bit about the vaccine design. That's also something that people are concerned about. So the adherence to continue these kind of prevention measures is definitely something of interest that some of you might be looking at the data for. Another interesting figure from the Pew Research Center, they have the vaccine interest broken down by gender, race, age, education, political party, family income. And then one point of concern here is just that Black Americans have been shown to be much less interested, much less willing to get a vaccine, and there could be lots of reasons for that, lots of racial reasons based on previous history with healthcare and um, healthcare institutions not treating Black Americans equally or appropriately. So there's still some fear and concern with different groups of people when it comes to these new vaccines. The mask issue was an interesting one at the very beginning of the pandemic. We now know very clearly, it's been demonstrated, time and time and again, that they do work. They are effective at preventing droplet spread, and those droplets again can contain the coronavirus and spread it from person to person. So wearing masks is quite an obvious way to prevent infection. At the beginning, it wasn't quite clear whether people should be wearing masks or not, but I think it came down to at some point it's spreading too rapidly, and people are getting really, really severely sick, that it's worth it to at least put in the time and effort to make sure people have masks on. And that's probably going to have to continue even now as we have two vaccines that have been developed. And then so the kind of last concern I wanted to touch on is one that we're still trying to figure out and trying to suss out. What are the long-term health impacts of having a COVID-19 infection? So here is a CT scan on the left, healthy lungs. Air is flowing just fine, freely. That person's breathing just fine. On the right are lungs of someone infected with COVID-19. As you can see, there is a lot of matter here, a lot of fluid, inflammation. Potentially that person is having difficulty breathing. And I've seen reports where people have seen lung damage that might be long-term, heart damage. People have self-reported that they've lost sense of smell and sense of taste. We don't know how long that might impact individuals that have infections. So there's still some things in terms of the long-term health impacts of having the infection that we're still learning about even now. So now to the vaccines, to the good news. So as I mentioned, the SARS coronavirus is very similar to this SARS coronavirus 2, which is causing the COVID-19 disease. And thus, You can imagine we've been studying a very similar virus from SARS since 2002. So we have a lot of data and information about that original SARS virus, and because they're so similar, it's applicable to this current pandemic. And that has helped really jumpstart the vaccine design. So typically, when you talk about vaccine development or vaccine design, you have these preclinical phases where you're identifying, what are the antigens or proteins that elicit an immune response. What are the pieces of the microorganism that we want to put into a vaccine that will help protect us from subsequent infections? That work was based on the work that was done with the original SARS virus. So we were able to jump into what you see here, which are the clinical phases, where you're working on um, patients, actual people, giving them different doses to see what dosage is safe, to see whether or not the vaccine is efficacious in actually protecting them either from an infection or protecting them from severe disease. And at the moment, you may know we have two vaccines that have been approved based on very similar technology by two different companies and consortia that have been working really diligently to get us safe, effective vaccines. So a little bit about vaccines. Typically, these four types of vaccines are usually what previous vaccines have been based on, right? So you have either an inactivated or killed bacterial cell or inactivated virus. You can also have a live attenuated. Attenuated just means weakened uh, vaccine, where you have the entire microorganism injected into a person. And because it's weakened, it can't actually cause disease, but it's still there and available for the immune system to see it, recognize it, cause an immune response, elicit an immune response that can then protect that person later on if they ever see the actual uh, fully strengthened microorganism. You can also have subunits or parts of a virus or a microorganism injected, and it's the same principle. You want to make sure that you elicit an immune response. Your immune response can learn and see what it looks like to see that microorganism, so that if ever you get infected or exposed, your immune system is ready, it's primed to respond rapidly and quickly. So the COVID-19 vaccines, both of them that have been approved thus far, are based on a different technology than most widespread vaccines of previous. They're based on mRNA technology. So in this case, instead of injecting a subunit or a piece of protein or a toxin or toxoid that's produced by a microorganism, instead of injecting that into a person to elicit an immune response, what we're doing is we're giving our cells instructions on how to make the important protein that our immune system can learn from and use to protect us in the future. So if you recall the central dogma of molecular biology, you typically can go from DNA. In our case, this is what our genome is made of, DNA. You have RNA or messenger RNA, which is what's used to express genes, and then those genes that are expressed can be made into protein. So we're giving ourselves mRNA that's specific to make the COVID-19 spike protein. We're giving our cells the instructions on how to make that spike protein. Our cells then make that protein and expose it to our immune system, which then learns what that looks like. They learn to react to it so that if ever we get exposed to COVID-19, hopefully we'll be protected. And so when I'm talking about exposure in vaccines, I'm talking about something that's depicted in this graph. So if you look at the y-axis, this is the concentration of antibodies. So as you go up the y-axis, you have more antibody response over time. So an initial exposure to any kind of microorganism, any kind of foreign antigen that can elicit an immune response. It allows your immune system to respond, but that response can be quite slow over time. And the number of antibodies that are designed to protect you is quite low in comparison to subsequent ex, uh, subsequent exposures. So that initial exposure takes some time to generate an immune response. And then that second subsequent exposure is a much quicker response. It's also a much more aggressive response. Your body is ready. It's seen that organism before. It's ready to fight and protect you. The vaccines are intended to mimic this initial exposure. They're intended to give your immune system an opportunity to learn what it is that they need to fight against, so that if ever you actually see the real whole virus, your immune system has memory. It's ready to protect you either against any infection or against severe infection. So this is actually a very exciting graph. This came from the FDA panel that met to approve, I think this is the Pfizer vaccine, the first vaccine that was approved a couple weeks ago. And so what they're showing here is the impact of this mRNA vaccine against COVID-19. The idea being very much like this graph, where if you can expose people with a vaccine, surely they should be protected and won't get the COVID-19 infection, or they won't get severe infections. And that's exactly what this is showing. So this is on the y-axis here is incidence, so the number of cases of COVID-19 over time is the x-axis. And what you expect to see, right, is people that get a vaccine versus people that get a placebo. The people that get the vaccine should not have many cases of COVID-19 compared to people that get the placebo. People that get the placebo will more than likely, if the vaccine works, have cases increasing over time. And that's exactly what is shown here. In red are the people that got the placebo. And over time, you can see that the number of cases of COVID-19, severe cases of COVID-19, increase over time, whereas people that got the vaccine stay quite low. And that's where that 95% efficacy that you might have seen on the news comes from. So this is a very efficacious vaccine. It seems to work at least at curbing severe infections in people. So even though we have two vaccines now, both based on the same technology, both seem to have at least 90% effectiveness at protecting people against severe disease, the pandemic still rages on. We have increasing cases, I'm sure, as you've seen in the news, an increasing record-setting number of deaths. And part of this seems to be attributed to holiday travel. People seem to also be dealing with quarantine fatigue. They're tired of having to stay quarantined and socially distance and wear masks. And so that might be some data and information that some of you guys might be researching and studying in your projects. We still have some remaining questions about these vaccines. They're new vaccines, but the data is very promising in that we think people will be protected quite well with these vaccines. There are some instances of adverse reactions or adverse events, most of those due to allergic reactions, which every vaccine has some allergic reactions. And in response, I believe the protocol involves people who might have allergies to take the vaccine, stay in the clinical setting or the hospital for 30 minutes, just to see if they have an allergic response. And if they do, they're going to be in the hospital. They're a good place where they can be treated, so that can be taken care of. So far, there are only a couple of adverse reactions, which is really good. We don't know the effectiveness of the vaccine in pregnant women. We don't know much about how much it protects or whether it's safe for children under 12. I believe only one of the vaccine trials included children. I think it was children from 12 to 16 or 12 to 18, I guess. But again, that was a very limited sample size. So there's still not a recommendation to have children getting these vaccines just yet. But the recommendation is that older people, people on the front lines who are at high risk or who might have chronic diseases, and then healthy adults are recommended to get the vaccine at the moment. And then there's, of course, this last concern that even if you have the vaccine, you still may be spreading the virus to other people. And the reason for that is because the vaccine is not completely protecting people. It doesn't seem like it's completely protecting people from getting any infection, but instead it's protecting people from getting severe disease. So you still might get infected after getting the vaccine, and you may be able to spread that infection to other people, which is why the continued use of personal protective equipment is still recommended after getting the vaccine. Okay, so this is my last slide, and I wanted to end on something that I think is really pertinent and relevant for you guys, which is that this pandemic, as any pandemic, is not just a biology problem or a medical problem. It involves lots of different disciplines, different areas of expertise. So we have people in public health and medicine, of course, but we also have engineers designing personal protective equipment, or designing the cold chain logistics. So the vaccine doses have to be kept cold. You have to figure out the logistics and engineer equipment to keep those doses cold so that they don't get inactivated or go bad. We have aspects of psychology and sociology, figuring out, whether people are going to continue to adhere to guidelines to socially distance, to adhere to mask wearing guidelines as well. We have aspects of economics and politics, computer science, analyzing all the data that's available. There's tons of data publicly available, which is a great thing, but figuring out what it all means, actually, that's your job to do. So putting all those pieces together is what's going to help us deal with this pandemic over time. And then so I want to make sure to end and let you know that your job is to really understand as best you can your piece of the puzzle, so that you can do the best work that you can do. And then also your job is to communicate what you've done to other people and recognize that others might be from different fields, have different expertise. They might be really focused on different types of problems that that means that they don't quite understand your discipline. So understanding and communicating what you're doing in your project is going to be really important. Okay, that is all I have. I don't know if we'll have time for questions, but I'm willing to answer any questions that might come up.
Yes, the question says why people with allergy sometimes get allergic reactions.
My understanding is that is mostly as a result of how the vaccines are manufactured. I know that I think these vaccines. I know that the flu vaccine uses eggs to be manufactured. I don't know if that's true for the COVID-19 vaccines or not, but there is a warning that if you have any allergies, and I think specifically egg allergies, then you should be careful about taking this vaccine. And again, it's just a process of the manufacturing process.
I guess then there's another question, say, would the mRNA vaccine better than other vaccines?
That's a good question. So the mRNA technology, I do want to mention, was first published in 1990. That was when it was first thought about. It wasn't perfect in 1990. They had to do a lot of work to get it where it is now. But the good thing about the mRNA vaccines is that they're easy to manufacture. So if you're trying to make a vaccine based off of an inactivated virus or bacterium or a subunit, you have to inactivate those viruses or isolate or clone those subunits. That takes time, it takes a lot of resources. On the other hand, creating a piece of mRNA is much easier, it's much quicker, and it just, as far as I know, it's logistically a lot easier to manufacture and distribute in the vaccine.
There's another question. How is the COVID-19 symptom different from the common pneumonia?
They are quite similar because they're respiratory infections. I don't remember specifically what the differences are. I'm kind of drawing a blank. But what I do know is that the mortality rate is higher for the COVID-19 infections. So the symptoms are actually quite similar. And as a result, using the masks and socially distancing to try and squash this COVID-19 pandemic, we're also limiting the spread of flu as well because it's spread in the same way. But as far as I know, the symptoms are quite similar, which is probably scary and confusing for people because you never know if you have flu or COVID or you have any other kind of respiratory infection until you get tested.
There's another interesting question. Will the COVID-19 vaccine also protective against the original SARS?
I haven't seen any. I'm not sure. I haven't seen any information about that.
I might jump in.
Yeah, please.
Yeah, if I might jump in. So not so long ago, we had Dr. Kizzmekia Corbett, who's coordinating the NIH vaccine effort, come and give a talk at Spelman. And what was very interesting there is that they actually started to develop the vaccine at the time where SARS were around. And so they looked at what were the different viruses that had pandemic potential. And one of the ones that was identified was a SARS-like virus. And so their strategy was to design a vaccine that would be universal for that category of pathogens, viruses. So it should be. I don't know if it's, obviously hasn't been tested, but the approach was to have a universally functional vaccine.
Oh, you're muted.
Oh, sorry. Someone raised her hand. I forgot where that person is. Can you type the question into the
That's
chat?
Haley.
I think I see her. Haley, I see you. Haley B. Yeah.
Hi.
Hey.
I would actually prefer to vocal my question if that's okay.
Oh, go ahead.
Thank you. Dang it, it just went right out of my head. Where did it go? Oh, that's right. My parents have been looking up some of the symptoms for COVID-19, and it's been very interesting because it's, I think it was the CDC is saying that there's some of the symptoms can be like headaches and diarrhea, and there's a whole lot of symptoms that are seemingly unrelated to a respiratory infection that are becoming related to the COVID-19 virus. Do you know why that's happening?
I don't. But what I think I know is that it seems to be kind of a systematic infection, systemic rather, in that it's impacting lots of different organs in your body. I don't know specifically why it might cause diarrhea or headaches. But I mean, those symptoms do make it very complicated to try and assess whether or not you have the infection. I actually thought I had COVID-19 earlier this year. Turns out I was negative at the time, thankfully, but that's why the testing availability was really important and still is, because it's really difficult to assess based on just symptoms alone, whether or not you have COVID-19. Dr. Ayeli, were you going to go ahead?
Yeah, I was going to add something to that. Is that so? Researchers, as Dr. Sistrunk just showed in her slides, the entry point for the virus is that ACE2 receptor. And there is this thought that it might be the availability of these ACE2 receptors that dictates which organs might be most affected. And so different cells have different levels of ACE2 receptors. And they even say that, the fact that children are not so much affected might be related also to their level of ACE2 receptors. So different organs have different levels of ACE2 receptors. But Dr. Coates was about to jump in as well on this topic.
Hello, everyone. So one of the things that's really interesting about respiratory viruses and SARS-CoV-2 is really starting to show us is that there's really evolution and natural selection on viruses. So in your respiratory and circulatory system is where most of your immune cells are circulating. And so no virus wants to be around the cells that are likely to kill it. And so as a consequence of that, there are certain sites in the body where you don't activate an innate immune response. And you don't activate that innate immune response in those areas, we believe, because the innate immune response is so non-specific. The amount of damage that it'll do just isn't worth it based on the number of times that you come in contact with different types of foreign pathogens, some of them that may not be as severe as SARS-CoV-2. And so those areas include the central nervous system. So you don't want to activate a non-specific response that may damage your brain or any of the nerves that you need, your reproductive organs, because as a part of biology, the ability to have viable offspring is something that's really, really important. And then also the eyes. So sometimes viruses will do their best to get to those different locations. And when they get there, they can have their own detrimental effects. And so in the case of SARS-CoV-2, what they believe happens is that the virus uses the circulatory system to be able to get into the brain. And then as a process of its replication, it releases reactive oxygen species that ultimately decrease some of the oxygen content within the brain. And so then throughout the course of the infection, which could be 2 weeks or more for different people, there's a reduction of oxygen in your brain, which can cause you to have that brain fogginess, as well as the headaches. And as far as the gut, it's also something that's evolutionary. Early on, a lot of the viruses and diseases that we got because of contaminated water or ill-prepared food. So within your stomach, there are actually gut-specific immune cells. And so what they've seen through some certain biopsies is that the virus actually will hang out persistently in the GI tract and actually cause those symptoms because of the activation of that immune response that kind of come with that stomach ache and that uncomfortable feeling. So a lot of this is just really just something that naturally happens with respiratory viruses, but just as a general population, it's been a really long time since the majority of the world has been impacted by a respiratory virus to really see these symptoms.
Harry Nelson.
I never heard of that.